Sorry, I was on mute.
Good
Evening,
evening.
Scott. Evening, Elna.
Hello, good to see you.
Hi, Chris. We
Yeah,
all
good to see you. Elna, I'm happy to do the introduction. I just need to find Scott's biography quickly. Did it come from
Chris,
Sunita?
I'm happy to do that. We already went live. The meeting
Oh,
is
okay.
live.
Oh, okay.
Just all.
Thank you.
um, everyone, thank you for joining tonight. We will just wait a few more minutes because there's still attendees joining us. I will start the introductions in a few minutes' time, and then we will hand over to Dr. Scott for his presentation. So just bear with us. In a few minutes, we will start the webinar. Once again, welcome and thank you for attending our first CPD talk for this year's educational webinar series. Uh, Dr. Scott Younger will be presenting to us today. Dr. Scott Younger is the Director of Disease Gene Editing within the Genome Medicine Center at the Children's Mercy, Kansas City. Since joining Children's Mercy, his lab has pioneered methods for the rapid, robust, and scalable production of patient-derived cellular models. The lab is currently focused on the development of platform-based approaches for functional precision medicine, with an emphasis on reduction to practical and clinical implementation. Dr. Younger came to Children's Mercy from the Broad Institute of MIT and Harvard, where his group worked on the development of new methodologies to expand the utility of CRISPR-based genetic screens. Prior to working at the Broad Institute, he completed his postdoc studies at Harvard University as an American Cancer Society Fellow. He holds a PhD in cell and molecular biology from UT Southwest Medical Center, and he also received a MS in biotechnology from the University of Texas at San Antonio and a BSI in bioinformatics from Baylor University. Dr. Scott will be talking to us today on scalable approaches for modeling rare disease and patient-derived organoid systems. I just want to ask you that you are welcome to use the Q&A as well as the chat functions to post questions throughout the talk. We will handle all questions at the end of the session. And if you also want to have an online discussion with Dr. Scott or you wish to ask your question in person, you are welcome to raise your hand and we will unmute you at the end of the session. Thank you very much, Dr. Scott. We look forward to your talk.
Okay, great. Well, thank you so much for the invitation to present in this series. I'm excited to share some of the work that's going on in my lab here in Kansas City, and all the work that I'll talk about today kind of revolves around patient-derived organoid systems. But before we get there, I kind of want to get kind of a foundation, the basics of biology, so genes and phenotypes. So for humans in each of our cells, we have 46 chromosomes. So we have two copies of 22 autosomes, and there are two sex chromosomes. And then distributed across these chromosomes is roughly 20,000 protein coding genes, right? And the spatiotemporal expression of these genes really gives rise to phenotypes. In the lab, we think about this a lot as cellular phenotypes, like cells in a dish, and features like proliferation. But cells make up our organs, and our organs make up our body. So the expression and function of these genes actually dictates human phenotypes. Um, and we saw this. This is kind of a really cool demonstration, I thought, that we saw very early on working in this space. And so what I'm showing you here, this picture, is what's called an embryoid body. And what this is basically a three-dimensional ball of pluripotent cells. And here you can see this nice tight ball, and this is from an unaffected individual. But when we generate stem cells from a rare disease patient, right, and we make these organoids, or sorry, these embryoid bodies, you can see right out the get-go. This is a rare disease caused by a single letter change, and we know this is causal. But even at the early stages in a dish of this embroid body formation, you already start to see these aberrations, right? And it really shows the developmental impact that genetic variation can have, right? And really, that's because our underlying genetics can inform, disease development or disease progression. And they also can dictate therapeutic vulnerabilities as well as responses to specific therapeutics, right? So baked into all of this is the underlying genetics. So it's, as many of you are probably familiar, we've been trying to generate cellular models of disease for a long period of time. I kind of have this picture I took from a publication. I thought it, best summed up kind of the current practices for generating models. Nowadays, if we want to study a particular gene or genetic variation, what we might do is use CRISPR-based approaches to engineer variation into cells, and maybe we make an animal model like a mouse, or we grow these cells in a dish. And this kind of approach to understanding genetics is genetic manipulation has really been mainstream since the late 1980s, and it's a lot more accessible than it was back then. Again, it's particularly with the advent of CRISPR technology, but it's still very time consuming, and it can be very expensive to generate cellular models this way, right? To model disease. And another, feature of this design is that it's a hypothesis driven approach, right? So you have to identify variation you're interested in and then engineer that into the model to study it. An alternative way of doing this is using patient-derived stem cells or individualized stem cells. And this is a process that iPSC reprogramming that was described in the early 2000s, or sorry, mid-late 2000s, right, where you can take a differentiated human cell, you could introduce specific reprogramming factors, and these transcription factors would revert those differentiated cells back into an induced pluripotent state. And then once you're back there, you can differentiate these in theory into virtually any cell type in the human body. So until recently, this has still been extremely time consuming, and it's very, very expensive. But the benefit you have here is this is ideal for discovery biology because the genetics are actually baked into the model, right? So now you can use that model that you've made to kind of parse out the features of the genetic variation that may be giving rise to a disease phenotype or in that cell model. So here I'll kind of talk about kind of where we're using this type of technology and how it dovetails into our rare disease initiative here at Children's Mercy Kansas City. So about 4 or 5 years ago, we launched our rare disease initiative, Genomic Answers for Kids. And it's a program where we're aiming to sequence the genomes of roughly 30,000 children impacted by rare disease. This is largely undiagnosed rare disease, right? And along with families to do trio sequencing and try and identify genetic variation leading to disease. So we early on thought, well, maybe these iPSCs, generating iPSCs from these patients would provide a nice tool for tying in the genetic variation that we're detecting by genome sequencing into cellular models. So the approach, in theory, is pretty straightforward, where we can just take PBMCs that have been isolated from an individual, right? And then we can go through this reprogramming step, and then we can get these induced pluripotent stem cells and then have a model for each patient that we can study in a dish. Now, at the time that we decided to pursue this, we actually didn't have any experience in this space. So our first pass was to reach out to contract research organizations to see if we could have this done for us. But a few years ago, when we started doing this, what we found quickly is that contract research organizations in the U.S., this process would cost upwards of $10,000. The turnaround time for many of the CROs was about a year. Again, this is per individual line, and several of these CROs actually had like waivers that you had to sign that said it might not work, but you still have to pay. So we're an academic lab, so we can't really pursue that kind of approach. So we brought it in-house, and through exhaustive and Herculean effort by a research scientist in the lab, Dr. John Means. He was able to survey the entirety of the literature, understand who was doing what and why, and what were the things that really needed to be happening. And he was able to reduce the cost of this process down to about $200 in reagent costs per line. and also do this reprogramming in a way that only required about 2 to 3 weeks. Another feature of the way that we've approached this is that we're working rather than getting fresh blood draws from patients. We're returning to cryopreserved blood cells that were collected when those patients enrolled in the program for genome sequencing. So we actually don't have a ton of cells for each of these patients. So we work in smaller plastics. So we actually do these reprogrammings in 12-well dishes, right? So we actually, it's really cost effective to do these, 12 at a time in a dish. So this is a figure from a paper we published last year, but this slide is constantly out of date. I think we're nearing in on 500 patient-derived iPSC lines that we've generated in our lab alone. So this plot right here, what I'm showing you is successes and failures in terms of reprogramming outcomes. Early on, we had some level of concern that there may be some intrinsic features of rare diseases or subsets of rare disease that would prevent the reprogramming step from actually working. But what we actually saw is there was a clear cutoff in terms of our failures. If we drop below 300,000 PBMCs going into their programming reactions, we actually saw that the reprogramming was less efficient. We revisited some of those patients with fresh blood draws and a full, million PBMCs, and they worked very well. So we know that really the critical feature is the number of PBMCs that go into the reprogramming reaction and nothing about any individual rare disease. So we've done extensive profiling on the iPSCs that are generating using this approach. So some of the things we've done is go through and look at pluripotency markers, and you can see that all of these canonical iPSC markers are expressed in these lines that we've generated. We've also gone through and we've done exhaustive genomic stability surveys of these lines, and you can see that they are in fact genomically stable. We've done this through lines, I believe, up through 30 passages. So we showed that you can really culture these long term and they retain genomic stability. And we've also assessed their capacity for differentiation. So we've taken these stem cells and we've differentiated down ectoderm, endoderm, and mesoderm lineages and verified that they are in fact pluripotent, right? So they have all the hallmark features of iPSCs. Great. And so the important thing to note is what can you do with these iPSCs and how are we applying them? Now that we've showed we can make these robustly, what are we actually doing in the lab? All of our work in the lab, or most of it now, is done in neuronal cell types, so neuronal lineages. So taking these iPSCs and making them into neuronal cells or cardiac cells. And so I'll show you how we're doing that. I'll also highlight that all of our work nowadays is done in three-dimensional organoids. So here's an example of a cardiac organoid generated from an unaffected individual. So if you're not familiar, organoids are three-dimensional cellular models that recapitulate organ development and function, right? So they're really valuable tools for studying development and disease. So again, this is a cardiac organoid from an unaffected individual. It will just sit in the dish and it will contract. But if you were to make a cardiac organoid from a patient with muscular dystrophy, what we found is that it actually just sits there. It'll occasionally kind of move, but there's clearly a contraction deficit. And this is remarkable because this is really what you're seeing is disease in a dish. So we can faithfully model human phenotypes, right, in these advanced cellular models. So for the rest of the talk, what I'm going to be telling you about is how we've harnessed this technology in the various spaces that we're working in. On the one side, part of my lab is doing precision and personalized medicine. And then the other side of the lab is doing more basic science, hypothesis-driven exploratory research. So I'm going to talk about kind of what those things are and where we're active in these spaces. But we'll start with precision and personalized medicine. So this is not a novel approach. So really this approach at scale was born out of the Human Genome Project, right? And so the idea that if you know somebody's genome sequence, that it would inform all of disease and also all of the therapies that could be used to treat. It's been a little more complicated than that, and that hasn't really borne out. But this approach actually has been successful in oncology-centric applications, right? Where you can sequence the DNA of tumors and actually identify mutations and use that information to guide therapy. So again, this is quite advanced in oncology, but this approach of using genetic information or genome sequencing to guide things has kind of been stratified into something we'll call genomic precision medicine. So the arm based on just genomics. What we're doing in the lab and what's really an exciting area right now is functional precision medicine. So that's where we're actually going to do experiments in a dish and get empirical data to inform treatment decisions. So again, in oncology-centric applications, you can envision taking tumor cells out of a patient. Then you can actually do drug screening in a dish or other assays that are going to say, okay, this is from that individual. We're seeing how that person's tumor is behaving to these drugs, so we know that this is the right treatment. Okay, so that's what we're doing a lot of in the lab in terms of that approach. We're not doing anything with oncology. We're taking these approaches and we're applying them to rare genetic diseases. Right. And so our big goal here at Children's Mercy, when we talk about functional precision and personalized medicine, is going from the patients we're seeing here in the hospital to getting cell models established in the lab that we can profile and perform rapid experiments in a timely manner that can actually inform the clinical care for that individual patient. So that's kind of the goal that we're working towards achieving. So I used two words here. I said precision and personalized. Those are similar but different, and I want to kind of clarify what those two different approaches mean. So in terms of what they have in common, the requirements, they require genetic or genomic data from a patient as well as biospecimens from that patient. The goals are the same in terms of finding a therapeutic approach for an individual. And the data we're using is also similar, genome sequencing or targeted experimental data. Now, so all those are the same. Where these differ is the actual therapeutic we're talking about. So for personalized medicine, we're talking about a tailor designed patient, a drug that's going to be for that one person. So this is what we call an N of one, versus precision, where this is taking existing and approved drugs and finding out of that category, what's the right approach or what's the right drug for this specific individual. So I'm going to start with where we're at in these areas with personalized medicine because we're a little more advanced in this area in terms of the preclinical side of things. So earlier on, I showed you this plot. What I don't want to get lost in this, right? This is a bunch of data points. But each one of these data points is an iPSC line that we've generated, and that was from one of our participants in our rare disease program. So that particular dot that I'm highlighting, that came from a patient with Duchenne muscular dystrophy that had a confirmed causal structural deletion. So this is a disease caused by mutations in the dystrophin gene, right? So this is a structural deletion, and so that actually that cell model I showed you earlier, those cardiac organoids, that was from this particular individual. So again, we see an unaffected individual, the cardiac organoid is contracting, and a cardiac organoid from this individual, we see that they're actually this contraction and somewhat of a structural deficit there. So again, this is caused by a structural deletion on the dystrophin gene. So this is a quite large gene. In this particular patient, they're missing exons 46 through 53. And what happens to the dystrophin transcript is it gets knocked out of frame. So in these patient cells, they're not making any dystrophin. This is an X-linked disorder. So predominantly occurs in males. Males only have one X chromosome. So only one copy of this gene. Now, one of the reasons that this particular structural deletion in patients stood out to us at the time was the FDA had just approved back in 2021 a drug was often referred to as an exon 45 skipping antisense oligonucleotide. So antisense oligonucleotides are short synthetic oligos, and you can design them to be complementary to RNA transcripts. They can influence gene expression and splicing. In this particular application, the ASO, as it's described, blocks splicing to exon 45. And the result of that is that now exon 44 will connect to 54, right? So you'll never get back the genetic information that's lost, but what you do get is a transcript that's been knocked back into frame. So that form of dystrophin that ends up being made has been shown to be therapeutically beneficial. So this provided a great platform for us to say, what happens when we take this drug that's been approved for humans and then we treat these cell models? Can we recapitulate clinical responses? So here I'm showing you, now this is immunofluorescence. We're looking at dystrophin expression in green here from a cardiac organoid. So you're seeing in an unaffected individual that green DMD expression, and in the patient, as you might anticipate, there's absolutely no dystrophin expression. And what we found when we introduced these ASOs or this ASO into the patient organoids, we see this restoration of dystrophin expression as we might anticipate. Now, we can go beyond this. And again, the powerful feature of the organoids is we can do functional profiling. So here I'm showing you a video of an organoid. So this is contracting. Now, the reason it's green is because these organoids have been soaked with a calcium indicator dye. So calcium release is part of the contraction process. So you're seeing this flux of calcium, and that's coupled with contraction. But when we look at this in patients with DMD, what we see is those organoids actually, they don't have that calcium flux. They're not contracting. You'll see an occasional burst of calcium, but clearly not functioning like an unaffected organoid. But when we treat with the ASOs, you can see you get this restoration of contraction, so functional restoration, and you see that recoupling with contraction. So a really cool example of how we were able to, model therapeutic responses in patient-derived cellular models. Now, this provided another opportunity for us to go back to our patient population, and I'm going to highlight another patient here. This is another Duchenne muscular dystrophy patient, but their cause of disease, the molecular cause, is different. This patient has a deep intronic variant in the dystrophin gene, and that activates a novel splice acceptor site and leads to the incorporation of a cryptic exon and premature transcript termination. So that looks something like this. So again, the end result is that no protein expression is made. Now, if we're thinking about antisense technologies, this is actually a great target for a personalized antisense oligo, because if we can target that specific variant site and prevent this splicing event from happening, we could actually, in theory, restore full-length dystrophin as a potential therapeutic. And that's exactly what we went after. So here you're actually seeing the sequence of the ASO. You're seeing it's complementary to the pre-messenger RNA transcript, and the ASO straddles right over that red nucleotide. That's the actual mutation in the patient. So we can do the exact same experiments that I just told you about, but this is in this patient, a different patient with this patient-specific antisense oligo, and you see again robust restoration of dystrophin expression. And when we look at the functional assays, we see again this robust restoration of cardiac function, right? So this is a really great platform for rapidly screening these personalized ASOs. Now, a big motivation for this report was the report published from Tim Yu at Boston Children's back in 2019, where he showed really the first soup to nuts patient customized oligotherapy. This was for a different disease. And that particular disease was associated with seizures. And you can see that intrathecal injection of the patient-specific or patient-customized ASO led to a significant reduction in the frequency and duration of seizures. So this was a kind of a approved approach that could potentially be pursued. If we look at that New England Journal of Medicine publication, you can see that process took quite a while, 16 to 18 months. And the preclinical side of things actually took, 9 to 12 months. So that was quite a bit of work. What we've been able to do is just for that preclinical component, we've been able to take patient cells, generate iPSCs in 2 to 3 weeks, generate organoids, cardiac organoids in about 2 weeks, and then run functional assays that take about a week. So really reducing that preclinical timeline down to about 6 weeks to do this preclinical leads. And we've done this for a handful of patients, and we still have some side projects with doing the preclinical component of trying to establish a potential for patient-specific ASO therapeutics. So I'm going to stop with that there and move on to kind of where we're working in the precision space, which is actually where we're really quite active these days. Now, for all of this work that I'm going to tell you about, we're doing patient-derived brain organoids. So we're still working with patient-specific iPSCs, but we're shifting our model into the neuronal space. So I like to show this picture just so we're all on the same page. A brain organoid, right? Again, it's a little 3D heterogeneous ball of neurons. It's about the size of a letter on a penny. And I show that this is not a sentient brain sitting in a dish, right? It's just a tiny cellular model. But we can make these. We've done extensive profiling to show neuronal markers. So this is a cerebral organoid, so or what some might call a whole brain organoid. So again, a heterogeneous mix. So when we take these, we can actually stain the organoids with that same flow 4 dye. So this calcium indicator dye. So calcium is also part of the action potential. So we can see bursts of green. Maybe you just saw it there when neurons fire inside of these organoids. So now we're in a microscope, we're zoomed in. Now, if we do the same process, but we generate the organoid from a patient with seizures, this is what we see. So we see this really hyperactive neuronal activity. It's a qualitative difference, just this extreme response. But really early on in these experiments, we were able to robustly see, and we've seen this for dozens of patients, that we can recapitulate this hyperactive neuronal activity for seizure disorders. Okay, so we're focused on trying to understand how we can apply these in a clinical space. This microscopy makes cool images, but it's time consuming. So we've adapted this to a 96 well plate approach, where we make our organoids in 96 well dishes, and then we can just apply these to a plate reader or set them in a plate reader, and we can actually read out neuronal activity. Here's an example of just like 12 wells from a plate. What we're looking at on the top row are 4 distinct organoids from an unaffected individual. The accompanying video is what you would be seeing if you were looking under a microscope. And then for the next 2 rows, these are 2 different patients with seizures. But what you're seeing across, again, 4 distinct organoids from each of those patients, you're able to see this really a qualitative difference in this hyperactivity. So this is really exciting because this is a much higher throughput approach to kind of screening through organoids and measuring that activity. And it sets us up to be able to do perturbations, right? And understand how we can treat these organoids and see how they respond to these perturbations. And perturbations in our case are going to be anti-epileptics. So here I'm showing you a plot it's kind of busy, but it's the same data I just showed you. Each box represents, an individual. And then all the different colors represent distinct organoids. So there's lots of replication here. So in the top box, you're seeing neuronal activity and organoids from an unaffected individual. And the bottom 3 boxes are 3 different individuals that all have seizures. Right. So now what we can do with this, in a single experiment, we can layer on a panel of anti-epileptics. There's roughly, over 30 of these that are approved for use in the United States. This is a really challenging treatment where only one or a small subset of these drugs work for a given individual, right? And it's nearly impossible to predict which one's going to work. So the clinical strategy is somewhat of a trial and error approach of put a patient on a therapeutic, stay on that for a few months, see if it works. You maybe change the dose, or maybe you switch the drugs. This can go on for many, many years, over a decade. And in many cases, patients end up with intractable epilepsy, meaning they've just no treatment. So what we've done here is seeing if we can model differences in patient responses to anti-epileptics. So here in patient 1, I'm going to show you 3 different drugs A, B, C. You can see patient 1 responded to drug A and C, but had really no response to drug B. And if we look at the other patients, kind of look at this matrix, you see patient 2 was B and C they responded to. Patient 3 was A and B. And I'll highlight the drugs A and B here actually have the same, they're different drugs, but they have the same mechanism of action. So on paper, you would anticipate that they would behave the same in patients. But what we're seeing is really this patient-specific responses to therapeutics. And this pilot experiment really inspired us to push forward with leveraging our rare disease program to generate patient-derived organoid models from patients with seizures, look at their clinical responses, and see, can we recapitulate that in a retrospective study? This is ongoing, but all results are very promising. I'll show you a snapshot here. This is a particular patient with seizures, again, part of our program. We can go back to that patient's electronic medical record. We can see the therapy or drugs that they've been treated with, and we can actually see how they've responded. I've highlighted 3 of these here because that's the data I'm going to show you today. So drug 1 on the top had no impact on seizures. Drugs 2 in red actually increased seizures. That's unfortunately not uncommon for this class of therapeutic. And then you can see in green, the third treatment will highlight has resolved seizures for 6+ years. So what we did is generate brain organoids from this patient and then treat them and then a dose response with these 3 different drugs. So on the left, again, you can see increasing doses really had no impact on seizure activity in the brain organoid. In the middle here, you're seeing that second drug. As we increase the dose, you're seeing a dose-dependent increase in seizure activity, which is what was observed clinically in the patient. And lastly, that third drug, you're seeing this dose-dependent resolution of seizure activity in the organoid. And so we can actually quantify that. And here you're seeing that kind of more graphically, where we're able to recapitulate the clinical responses. We've done this, in nearing a half dozen patients and seen a really nice correlation with what was observed in the clinic versus what we're able to rapidly observe in the dish. So, while we were in the process of doing this work, kind of to show the kind of the pace at which we're working and our interaction with our clinical partners, while doing that work, I got an email from a neurologist here regarding a patient with a CACNA1C calcium channel mutation. So this is an ultra rare disease and a suspected a gain of function variant that may be causing. Now, they were interested in seeing, could we try and establish an individualized therapy for this patient? So this was on June 29th of 2024. And so what we were able to do was get a blood draw from this patient, generate stem cells, and then generate brain organoids in a process that required about 8 weeks, which was sufficient time for us to design a panel of allele selective antisense oligonucleotides, again, tailored to the gain of function mutation for that patient. And here's the data that we were able to generate by August 5th of that year, which is exactly 8 weeks and 1 day. The data is plotted a little differently, but you're seeing the same thing, this hyperactive neuronal activity in an untreated organoid. NT is a non-targeting control or negative control ASO. And then for the allele selective ASO, we see this significant resolution of the seizure phenotype, which both validates that the variant is in fact gain of function, and also that this ASO could potentially be an individualized therapeutic. Now, the path from going to preclinical data to actually treating a patient is a challenging one on many fronts in terms of the regulatory aspects, which are changing for the better in this particular application here in the US. But the cost is still there, and getting through that process is extremely challenging. So one of the things we decided to do in the same experiment that I'm showing you here is we also tested a panel of calcium channel blockers because we suspected that this patient's seizures were being caused by this overactive or hyperactive calcium channel. And we do have FDA-approved calcium channel blockers. Now, these are not approved for seizures. These are hypertension medications that are typically prescribed in adults. But we went ahead and in the same experiments, we tested these 3 compounds, and we found one calcium channel blocker, nifedipine. It was actually a really potent inhibitor of that seizure activity. And the clinician took this information and along with lots of consultation, they administered the drug to the patient. And here's an email from the mother of the patient. Again, this is 3 months after we first learned of this patient, and the patient was no longer presenting with outward, presentation of seizures. So we were able, in the course of 3 months, on the margins, essentially, able to individualize or individually repurpose a drug for this specific patient. And this is where we're focused moving forward in these type of applications. Now, I told you about seizures, which impacts about 470,000 kids here in the U.S., but we're also interested in expanding our applications into other neuronal conditions, including attention deficit hyperactivity disorder. There's also indications from other labs, not ours quite yet, that you can model extreme depression phenotypes in these organoids. And we actually have other cardiac programs where we're modeling cardiac arrhythmias in addition, looking at antiarrhythmia medications as well, right? So really expanding the breadth of conditions we're able to profile and screen. We're also evaluating new technologies, the multi-electrode array machines. So this, rather than using a calcium dye, we can actually set our organoids down on a specialized plate with these semiconductor microelectrodes and actually read out electrical activity off of the organoids directly. So we're experimenting how we can put that into our workflows, and that's exciting. But I won't show any data from that today. So now kind of taking a turn into the other side of our lab. All that work I showed you previously is how we're trying to build up individualized cellular technologies for clinical applications, but we also kind of want to do research and you leverage these models to learn more about rare disease and how we can diagnose these and how we can study genetic variation. So one of the things we're doing, which is an NIH-funded project, is understanding or performing, systematic identification and phenotypic characterization of genetic variation, specifically for this grant study disease-associated birth defects. The idea here being we can take iPSCs derived from individuals impacted by specifically structural brain abnormalities and generate organoids and understand how development occurs in the dish and see if we can understand the molecular underpinnings of disease. Here's kind of just to show you at a first glance, as we're generating this data, you can actually see these changes. So this is two replicates of a brain organoid from an unaffected individual. And here I'm showing you from 6 different individuals, brain organoids that we've grown from patients with structural brain abnormalities. Now you can see it's not that there's not perfect replication in between the replicates, but you see this consistent structural abnormalities in the actual organoids themselves when we derive these from patients that also have physical structural brain abnormalities and observed in the patient. So we see this structural phenotype. One of the things we're moving forward in the next 18 months into performing spatial transcriptomics, where we're going to be taking these organoids now that we know have structural phenotypes, and then slicing these. We layer these layers down on a specialized puck that has oligos arrays. So it's basically like single cell sequencing, except because the oligos are fixed to the puck, you can actually get spatial information so we can look at the differences between development. So like I said, it's kind of like single cell, but we'll see how these organoids develop structurally or their cell compositions are different across rare diseases. Another area that's been quite exciting is, again, studying these seizure disorders. We were really interested in kind of tying the applications I told you about earlier together. We knew we can target splice disrupting variation as therapies, and we knew we could model seizures in a dish. Can we combine those two things, right, and hunt out rare genetic variation, specifically splice disrupting genetic variation, and hopes of using that as a handle to identify or develop preclinical leads for patients in our program. Again, this is just 6 different patients that all have seizures. These are zoom in on brain organoids generated from these 6 different patients. And again, just to highlight the reproducibility of this neuronal hyperactivity phenotype, right? So we know this is really robust. So the idea of this kind of project, this is kind of this whole full stack therapeutics of taking a patient. This first stop, we're going to perform a whole genome sequencing to get variant identification and curation. The orange is a process where we can actually screen through variation and identify those that disrupt splicing. I'll talk about that in a minute. The green, we can actually model diseases of unknown function in zebrafish through a collaboration we have, so we can do that quite rapidly. And then once we have a splice disrupting variant that we know is associated with a condition, we can generate brain organoids. That's the blue stop here, and purple generating patient-specific ASOs. So kind of this full circle. And in light gray, that last connection back to the person is that one day potentially have this as the full cycle of generating patient-specific drugs. But back to that functional profiling assay, if we have tens of thousands of patients that we've sequenced, they're undiagnosed, meaning we can't find obvious genetic causes of disease. can we develop tools or build tools and technologies that will allow us to survey intronic variation and help us to identify variants that have been missed in genome analysis that may be altering splicing and leading to disease, right? So intronic space makes up about 35% of our genome, so it's a pretty wide area, and if we can find genetic variation in there, we know what the impact is, we know how to target it. So in order to find these, we built up a mini gene splicing assay. So this is pretty straightforward. It's just a GFP reporter gene that we've broken apart into 2 exons. And we've done that by cloning in a short human intron. Right? And in the middle of that intron, we've put a cloning site where we can drop intronic sequences of interest. Right. And so we're focused on sequences that are about 100 nucleotides in length. Right. So if we drop in a sequence of 100 nucleotides and we try and profile splicing, if there's no impact on splicing, the GFP reporter gene is going to splice just fine. The cells are going to grow green. We can PCR over the exon-exon boundary and see a nice clean PCR product. However, if the sequence that we drop in there does impact splicing, right, it's going to disrupt GFP expression. And also, if we PCR over that boundary, we're going to see some kind of hybrid PCR product, right? And then we're going to know that splicing was disrupted in that reporter. Okay. So this works really well in cells, and we've adapted it to a high throughput format, where rather than profiling one sequence at a time, we actually profile tens of thousands of sequences simultaneously. And the way we do that is we print up on an oligo array, tens of thousands of sequences. Those are chemically digested off the array. We PCR them up, and then we clone them into the reporter gene. So now we have a plasmid reporter pool that has hundreds or tens of thousands of different sequences we're profiling. We can take these and transfect them into cells, right? And then we've adapted this to a multiplex barcode readout system where we can PCR over the exon-exon boundary and then use next-gen sequencing to read out splicing outcomes, to basically determine if splicing was impacted and how splicing was impacted. So here's an example of a single experiment where we're able to profile, right, 4,600 different variants. And so this for each variant, you have the reference sequence and the variant sequence, and we also have, I believe, 10 different barcodes for each sequence. So you can see how this 4,600 variants actually translates to about 92,000 different sequences we're profiling. And in this plot, what we're doing it, we looked at a very rudimentary splicing metric for splicing outcomes. And basically saying between the reference allele and the allele with a variant in it, again, the variant coming from patients that we've detected inside of our rare disease patient population, right? What are the variants that alter splicing? The red dots are these outliers. So these are variants that caused either splice acceptor sites or splice donor sites to occur in the reporter gene. Now, in this assay, we spiked in some controls. So here's 3 different variants that we spiked in from the literature. These are all confirmed causal for rare diseases. And we saw that these 3 different variants all cause significant splice disruptions. But what was remarkable, we didn't anticipate in our assays, if we look at the actual sequencing data, we saw that these splicing variants that we were detecting in our splicing assay were accurately recapitulating the exact splice disruption that was observed in the patient, right? And so that top variant that I'm showing you in this list here, that's the actual DMD variant from our patient that I showed earlier. And if you're familiar with the splicing, you may know that on the intronic side of a splice site, canonically, you'll see an AG there. So you can actually see that red nucleotide, that G, is activating that AG, and the blue is the new cryptic exon. And the same for these other 2 variants. You're seeing how they're activating that AG. But what we were also able to see is these novel, splice sites that weren't from the activation of the splice acceptor site, but actually either upstream or downstream of the actual splicing event, suggesting that they were turning on or activating splicing enhancer sequences or splicing silencer sequences. So we're able to get single nucleotide resolution on how these variants are actually giving rise to these novel splice sites. Okay. So one of the things we first wanted to do, this is, It's not a trivial experiment to do, so we wanted to make sure that we needed to be doing it in the first place. So we took our empirical data from profiling these rare variants and their impact on splicing, and we compared them to um, these AI-based splicing prediction algorithms. So here's one of the main ones here, Pangolin, that's widely accepted. Now, if the AI-based predictions and then the empirical data matched, you would expect there to be a line blazing down the axis. But here we see very, very poor correlation between these prediction methods and the actual experiments. And that's not to say that either one is right or wrong. They may just be detecting different things. But we always err on the side of empirical data when we're making decisions on what to pursue. I'm going to talk a little bit about one of these variants. So again, each dot here represents an intronic variant from one of our patients. So again, this is on chromosome 16. This is in this gene ANKRD11. And here I'm showing you the clinical presentations from the specific patient which this variant was detected in. Um, and highlighting specifically that this patient has seizures. So we have a functional assay that we know we can measure, and also hypoplasia of the corpus callosum. That's a structural phenotype that we believe we can model in a brain organoid as well. So again, so the idea here for this predicted variant, a splice disrupting variant, is it would behave very similar to the DMD model I showed you early on, where it would be disrupting gene expression. And sure enough, if we make an organoid from this patient, you can see ANKRD11 expression and brain organoids from an unaffected individual, but you can see this heavily reduced expression in organoids derived from this particular patient. Right. And we also see when we make brain organoids from this individual, we also see that there are structural anomalies in the patient-derived brain organoid. And then looking at the function of these, we can see again, in an unaffected organoid, you see this occasional blipping of a neuron firing. But when we make organoids from this patient, you can see this significant neuronal hyperactivity, right? So we're able to model the clinical presentations directly in a dish, which is promising. And here's this kind of quantified on a plate reader assay here. We also, as I mentioned, that gene has some associations with disease, but we wanted to validate that gene did cause phenotypes. So we knocked it out in a zebrafish. And what we're seeing here is there is a developmental phenotype in the brain of the zebrafish. And so you see this overall reduced brain size, which the particular parts of the brain that are reduced match kind of the structure of the zebrafish brain that would map to the corpus callosum, so that tracks. And we also see this functional assay where in a zebrafish, the touch response, you can tap it and the fish will move away. But in this fish, when you knock out ANKRD11, you see kind of this spastic behavior, which has been used to model seizure-like or hyperactive neuronal phenotypes. So all of these things show that when you knock that gene out in a model organism, it matches the clinical phenotype. So coming back to our patient, we can actually design a patient-specific ASO, right? You can see that with the one we designed here, it straddles right along that U. This was a tough one to get because right upstream of the variant is a poly-T track in the genome. So there's not a lot of sequence specificity for designing a patient-specific ASO, but we were able to get one. And here you can see when we treat the organoids with ASO to prevent, trying to prevent the splice disruption, what you see is this a restoration of protein expression, as we might anticipate. And then again, we also see a resolution of that phenotype, right, of the neuronal hyperactivity. So again, this looks to be another example of this patient-specific ASO. Now, in terms of our other projects that kind of match this in similar spaces, we're doing a lot of rare disease research focused on assistance with diagnosis. Again, that example of the patient-specific ASO I just showed you, that was not necessarily just a therapeutic. That also was used to validate the function of that variant and tying that to its relationship to the patient's disease and trying to achieve a diagnosis. These are all internal collaborations we're doing with clinical pharmacology to look at patient-specific responses to ADHD medications. We're doing a couple of different projects with different viral infections. So in some of the patients that are seen in the hospital, HSV or RSV, they can lead to encephalitis and things in a very, in a small subset of these patients. And getting blood draws, making organoids from these patients and trying to understand, can we model that phenotype in a dish and better understand how to predict when that phenotype is actually going to happen in the clinic. And then we also have ongoing collaborations looking at oncogenic transformation, in pediatric cancers, and can we show that or model that in patient-derived organoids or model that disease development. So today, overall, I've shown you kind of a summary of the platforms that we've developed here, which is focused, anchored in our ability to do these personalized cell models where we can take, PBMCs from a patient, rapidly reprogram those into stem cells that we can biobank, and then we can do characterization on those. One arm of the lab is focused on personalized therapeutics. So how can we then take these models and use them to generate or develop preclinical data, preclinical leads for compounds, individualized drugs that may one day be used to treat these patients. And also precision therapeutics, where we're really, really excited and active about where how can we implement this clinically where we can actually start to predict drug responses in a timely manner, right, to reduce the time to treatment for our patients. And in the party few minutes, I just wanted to highlight kind of one last area that we're excited about, which is the reassessment of abandoned preclinical leads. So this is actually a story that started long ago. This was my graduate advisor, and this is a project that was actually done after I left the lab, so I had nothing to do with this project, but a project in David Corey's lab where they were looking at ASO methods for trying to approach Friedreich's ataxia. So this is a rare disease caused by an intronic repeat expansion within the frataxin gene. And as that expansion gets longer, all of a sudden the protein is no longer made, and that reduction in frataxin protein is what causes Friedreich's ataxia. So in David's lab, one of the things they did was design these ASOs that were targeted to the specific repeat. And what they were able to demonstrate in cellular models was that these ASOs could actually restore frataxin expression, and they were able to show that in various cell types. So really promising lead. But one of the things that happened was when they moved into animal models of disease, specifically mouse models, they found that those ASOs did not work. And they responsibly wrote, a manuscript trying to describe the difficulties between the human cell-based models and the animal models and why that might not have worked. But just a few years ago now, the FDA here dropped the requirement for animal testing before human drug trials, which kind of raises the question of what would happen if these experiments were done today, and if the mouse is no longer required. Is this drug or could this drug potentially be a good candidate and may have been overlooked? Now we happen to have Friedreich's ataxia patients here in our rare disease cohort. I'm going to show you one here. Here I'm showing you the pre-mRNA transcript. You can see this is the repeat expansion that leads to disease. So we were interested in what would happen if we actually took these ASOs that had been tested in kind of standard cell models, and we actually use them to evaluate, their impact on patient-derived organoids. So here I'm showing you brain organoids from an unaffected individual in the top row. In green, you're seeing frataxin expression. If we generate these organoids from the patient, you see, again, that's a non-targeted ASO. You see there's no frataxin protein expression. But when we treated with these ASOs, we saw this remarkable restoration of frataxin expression, which is what had been observed earlier. Now, these organoids allow us to go into a little bit more detail in the function. So here now I'm looking at this neuronal activity assay like I've shown you before. We can see kind of the regular amount of activity we see in an unaffected organoid in the top row, and then that neuronal hyperactivity, which we see in ataxia. Again, so similar to what we see in seizures, we see the same thing for ataxia. So that hyperactivity. And here in the bottom two rows is, a non-targeting ASO versus the repeat targeting ASO. And here you're seeing that repeat targeting ASO actually significantly decreases that neuronal hyperactivity. So we see a correction there. Now, this is also another unique case where these patients also have cardiac phenotypes. And what we see in cardiac organoids from patients with Friedreich's ataxia is we see contraction again. So this is, the periodic contraction. The top row is what we typically see of an unaffected individual. And then that second row, you're seeing this weak but rapid contraction. But when we treat with the ASOs, we see these things snap back to that strong periodic contraction. So we're seeing in multiple systems that these ASOs actually restore function. And I should say we've seen this across 4 different patients, so this is consistent. And that varies with the number of repeats, right? The other thing that I think is important to note, I don't have the data to show you here, but to highlight the impact and power of these organoid models is we've seen that you can actually take the stem cells from these patients and treat at the stem cell level with the ASOs, which restores frataxin protein in the iPSCs. And then if you move forward with the differentiation with frataxin restored, you actually get what's seeming a totally normal development of the organoids, meaning they never develop these phenotypes, suggesting that these might also be tools, strong tools for not just looking at therapeutics, but also for profiling and understanding disease development. So I think we're right on time. So just a few acknowledgments. All of the organoid work I showed today was performed by Dr. John Means in the lab. The splicing assay that I showed you was developed by Annabelle Martinez. A lot of this work was done in collaboration with Genomic Answers for Kids, led by Tomi Pastanen. And then the fish work was done with a local collaborator at the Stowers Institute, Paul Trainer's lab. We're funded by the NIH and local organizations, BioNexus KC, and of course our hospital. That's an image of our hospital. Please look us up. Please advance on our website to learn more about what's going on now. And with that, I'm happy to take any questions. So thank you all very much for your time.
Thank you, Scott. I'm as expired, if not more, than what I was when I heard you in Mayo last year. So yeah, I think my experience in the last 15 years is, in South Africa, there's been a lot of improvement in our diagnostic capability. We diagnose a lot more rare diseases compared to what we did 15 years ago when I started in really inherited metabolic diseases. But what I've realized over the last year or two is that, in fact, all that we've done really is to move patients from the diagnostic odyssey to the treatment odyssey. And I think the work that you have shown us provides some kind of hope, or not some kind of hope, provides hope that, that much more treatment options can become available for patients in the near future. I see there's no questions at the moment. Maybe people were a little bit overwhelmed. I was certainly overwhelmed when I listened to you the first time. So I think I'm going to start off and just ask you a few questions. And then while we give people a chance just to formulate their questions. Well, Elna's just told me that there's already actually questions in the chat. I didn't check the chat, so let me just go there. Okay, so Glenn Kumalu is asking, I was wondering whether the ASO treatment can be applied to loss of function variants or if they are generally limited to gain of functions.
Yeah, so gain of function is definitely the more obvious approach for using this. Loss of function can be addressed, but that's a little bit counterintuitive in terms of, for example, the DMD of the splice switching. So we're working on another one of those now. We're trying to basically skip over exons that have loss of function variants or something like that to restore some altered version of the protein, or alternatively activating orthologs or things in a way. So those are a little bit more indirect, but those approaches are out there, but they're just not as tailored. The gain of function is a more obvious approach.
Okay, and then the last time when I spoke to you, you said that there was plans to publish some of your protocols, and that in the near future, I think you said that we may be able to read some of these protocols. Have they been published or not?
Yeah, so near future, it's funny how we're always optimistic about that. We're in the process of late in the process. I'm hoping near the end of a process of doing a protocols manuscript that's going to have all of the iPSC derivation details in there. Again, so there's nothing proprietary there, A lot of the details, we published that paper in Nature last year that had a lot of the details. The protocols paper is going to be a little more granular in terms of little steps and tips and tricks in there. I'm optimistic that'll be out in the next couple months and have more of that detail. But again, there's nothing proprietary there. It's just kind of more of the optimization of the approach that we'll be able to share more details about. So hopefully soon.
Okay. And then just another question from my side. So, I mean, in our laboratory, a lot of the work that we do is in inherited metabolic diseases. So the organoid would be liver in this case, and often it's metabolic pathways and defective enzymes. So, besides the obvious that's been used for a long time, substrate and product measurement, which has got its own challenges, is there any other reporter that you would advise if one had to study liver as an organoid?
Yeah, I mean, I not at the moment. So we're doing some of these. One of the things that we're dabbling with is with the brain organoids, using mass spec based approaches to look at neurotransmitters and things. So you can envision if for a liver organoid or something, look at analyze other things in there using mass spec based approaches, or really we've focused a lot, hopefully it comes through on scalability. So we've adapted pretty much everything to plate reader based assays so we can rapidly screen through there. So if there is any, specific enzyme function assay that can be adapted to that format, that's kind of on a use, case by case basis. I don't think there's any perfect like out of the box approach for some of that.
Okay. And then maybe just in terms of the N of one studies, what need to change? What changes is required preclinical? I mean, I actually attended a presentation from Timothy Yu about the development of Milasin, and it was incredible how quickly they did it, but it was still too slow, So what needs to change so that this can actually become an effective approach?
Yeah, I think this is a really tough challenge. That was a really, really strong proof of principle. You've seen what can happen when you get all of the best people together, firing on all cylinders towards a goal. So it's doable. The approaches that are going on now are pretty conservative in terms of the DMD patients, for example. Those patients will never see that ASO. And the reason is that it's too expensive, right? So nobody can foot the bill for that. The regulatory burden is somewhat dropping, but for DMD, for example, that is a degenerative disease, right? So it's
Yeah.
one where the patients are not, at the critical stages of diseases just yet. So how do you give an experimental drug to somebody who doesn't maybe have the extreme symptoms? And so that gets kind of a kind of a gray area we don't have good answers to. And for the conditions that are being pursued, there's still this abundance of data that is required in preclinical data. Aside from what I've just showed you today, people are still wanting to see animal models. And an example we tried to pursue, for example, that the seizure personalized ASO we pursued, we showed that worked really well, uh, but there was still push to get an animal model to show the exact physiology of the channelopathy, right? So it's kind of one of those things where the burden hasn't dropped to a point, even though we can generate this kind of data relatively quickly. And that's actually a lot of our work in the precision medicine space is because of these hurdles. And we saw that we just couldn't get past any of those and getting towards a patient treatment. Whereas taking the same tools and applying them with established drugs, we could get right into the clinic. So I still have hopes for the N of one, but I think that path is still it's just a challenging one.
Okay. Sizwe Chabalala is saying, thank you for the great talk. I'm interested in your iPSC reprogramming approach. Sizwe, I'm also interested in that. We currently use Sendai virus to introduce the reprogramming factors, and that takes approximately 1.5 to 2 months. I'm interested in how you managed to shorten this to 3 weeks. Quite incredible.
Yeah. It's really, I mean, that was a tour de force by Dr. Means in my lab, and really it was just process improvement. And this is an episomal nucleofection, so it's far less efficient than the Sendai virus. But the reason we're doing that is because it's also far less expensive. We couldn't afford to do Sendai virus on all of our patients. Um, but real, it's PBMC isolation, nucleofection and plating, and it's just the medias that are used in there that help this process go along. One of the things I'll highlight, we do polyclonal reprogrammings. We don't select clones. That's something that one could do if they wanted to. We find obviously it slows down the process, and you risk picking a clone with an artifact. So the population approach speeds things up. In a way, if you're trying to get a clonal line, that's still going to take you longer. But all those details will be in that protocol once we finally get it out.
Okay. I see no more questions, and we're slightly over time already. Scott, thanks again. It was a great presentation. I really enjoyed it. I feel really inspired. I'm pretty sure everybody in the audience is also inspired, and we look forward to see what's going to come from your lab in the near future. Thanks again. We really appreciate it.
Right. Thank you. Thank you so much for having me. I really, really enjoy doing this.
And then just quickly for our audience, thank you for your attending. The next CPD presentation, Elna, just correct me if I'm wrong, is in 2 weeks' time. Or is it next week?
No, it's in 2 weeks' time.
So the next presentation is in 2 weeks' time. We look forward to seeing you there, and have a pleasant evening and good night. Thank you, everyone.